Hi there, my name is Harikrishnan and
I'm Ryan Conley, and this is our report on RNAi and C.Elegans lab.
the overall purpose of our lab was to use the RNAi system to identify the function of a specific gene that is silenced in our C.Elegans.
Our research started by recording appearance and behavior of the RRF wild-type C. elegans. The C. elegans were in a petri plate that contained NGM light agar with OP50 nematode growth bacteria. The wild-type worms were most likely hermaphrodites and moved in an S-like pattern and also moved fairly quickly. They are always moving and rarely staying stationary.
Here you can see an abundance of worms in all of their stages. The egg, L1, L2, L3, L4, and the adult. After observing the wild-type C. elegans, we carefully transferred them using the chunking method into a new agar plate with the double-stranded C. feeding bacteria. For the chunking method, we carefully lifted a small cube-shaped chunk of agar and placed it upside down into a new plate containing the bacteria C.
After a few days of observations, we noticed a difference in their movement. The C. elegans no longer moved in the S-like shape they had before. As seen in the video, the C. elegans began to struggle with movement, such as moving back and forth, putting themselves in a U shape. They attempted to move, but instead of moving like they naturally do, they ended up rolling over when they moved forward, which caused them discomfort, so they stayed back and repeated the same process.
The gene that caused this behavioral difference is the roller phenotype, ROL6, and this gene encodes for the protein collagen. In C. elegans, collagen is a structural protein that keeps the body intact. The protein plays a critical part in locomotion. Without this gene, the C. elegans will not be able to move as quickly and freely as before.
The RNAi system is found to have control over the genes that are active and how active they are. This system is an important immune response to viruses or other foreign RNA. First, the double-stranded RNA entered the cells of the C. elegans as they consumed the bacteria. The dicer recognized the dsRNA of the bacteria to be possibly viral or foreign and cut into small interfering RNA, otherwise known as siRNA.
The siRNA then binds to several proteins, making a complex known as the RNA-induced silencing complex, also known as the RISC complex. Like DNA replication, the double-stranded RNA is then unzipped, leaving a single strand of RNA. The RISC complex then probes the cell looking for that specific mRNA with the specific nucleotides until it finds its match. Now the mRNA found can no longer be translated into a protein. Therefore, the gene has effectively been turned off.
Our overall impressions with the lab is that we thought the process of silencing a gene was very interesting. The process of determining what the gene did for the C. elegans was a fun process that included critical thinking, observations, and research.